So I'm excited to present to you today on how you can scale up your experiments with sample multiplexing on CyTOF technology. CyTOF is based on mass spec, which detects the mass of that metal isotope tagged to the antibody. Using these stable metal isotope tags enables many benefits. The discrete signal from these metal tags with CyTOF also enables multiplexing, aka barcoding. So barcoding applies a specific metal key to each of your individual samples. So here we have samples individually shown. Each of these would receive a specific, unique metal key. This enables us to pool these samples together as one large sample and then stain and simultaneously acquire that large sample. After acquisition, you would then debarcode your samples for analysis according to the assigned metal key. So why should we barcode? Primarily because barcoding greatly improves data consistency and reduces variability. It reduces sample-to-sample variations due to technical differences because the samples are stained and acquired together. It also improves single cell identification because with barcoding, we're using a specific metal key of 3 isotopes to identify a single cell. And we can see this demonstrated in this publication where they have used healthy PBMCs that are either barcoded as one large sample or individually stained and acquired. And we can see the median signal intensity CV% are dramatically lower for the barcoded samples compared to the individual samples. Barcoding can also greatly improve your workflow efficiency. It can reduce your sample prep time, acquisition time, the staining reagents needed, and it can also reduce the cell loss during staining and acquisition. So barcoding is really a great strategy to utilize for scaling up your experiments. Barcoding is a very well-published technique, and it has been in use for over a decade with more than 500 applications. You can use barcoding for a variety of reasons. For example, you could use it before and after a treatment in the same patient. This was done in this phase 2 metastatic breast cancer clinical trial, where they paired PBMC samples before and after a treatment. Barcoding applies to many species, including humans and rodents. In this application, they used a neuroinflammation, multiple sclerosis mouse model, and they barcoded mouse, spinal cord, and PBMCs into the same tube. Barcoding also can utilize many diverse sample types. This includes tissue that may be from a subject, cell lines, cultures, or even organoid models, such as in this publication where they're looking at colonic mouse organoids. There are 3 workflows that you can use with barcoding. Each of these really differ in where you place the barcoding step in the protocol. The first workflow would be barcoding before staining and acquisition. So here, your samples are barcoded, each with a specific, unique metal isotope key. They're pooled together, and this is followed by surface staining and intracellular staining, if that is applicable to your experiment. This is followed by acquisition and debarcoding. The second workflow is a barcode in between staining steps. So if you have surface and intracellular markers in your panel, you would first surface stain with your surface panel, barcode and pool your samples together. This is followed by intracellular staining, acquisition, and debarcoding. The third workflow is a barcode after staining, but before acquisition method. In this method, we would stain with both our surface and then intracellular antibodies. Then we would barcode the samples and pool them together to acquire as one large sample, followed by debarcoding. So now that you understand these workflows, let's discuss some experimental considerations for barcoding, and then I'll talk about how each of these considerations apply to the options we offer. So one consideration of your experiment will be where exactly in the protocol to barcode. This is the exact same workflows that we just reviewed, and which workflow you pick may depend on these other factors here. For instance, compatibility of epitopes may influence which strategy you choose. Some strategies require fixation before barcoding. Thus, if you have fixed sensitive epitopes, you would want to stain for those markers before fixing and barcoding. You may also want to consider what cell types you're labeling with your barcode. Some strategies label only immune cells, while other strategies label all cell types, meaning they are cell agnostic or universal. The number of samples you'd like to barcode can also impact your decision. Some strategies barcode more samples, meaning they're higher plex, while others barcode less numbers of samples. Another major consideration is optimization of the barcoding step. Just like antibody staining, barcoding is another step in your protocol. So barcoding is a step in your protocol just like staining and does need to be optimized for. So titrations should be performed of the barcodes. Your cell counts should be as consistent as possible from sample to sample. And keep in mind, sample quality can impact barcoding. If the barcoding step is not optimized for properly, this could result in not being able to properly identify your samples with debarcoding. So pilot studies for optimization are absolutely necessary. Finally, while barcoding dramatically reduces variability, it doesn't equal perfection. There are always other sources of variability that we as scientists should consider. For instance, variability could also be reduced by freezing your antibody cocktail for your panel and ensuring that your sample has high sample quality. Standard BioTools offers 3 barcoding options. This includes the Cell ID 20-plex palladium, or PD, barcoding kit, CD45, and T-mol barcoding. So I'll dive in to each of the considerations for these strategies. For palladium, the barcoding key is shown here, which uses palladium isotopes in the mass range of 102 to 110 PD. This kit comes with 6 isotopes, and you choose 3 to label 20 samples. So the nomenclature for this is often shown as 6C3 or 6X3. Palladium barcoding is currently the most streamlined option. It's likely the simplest, and it's a great place to start if you have never barcoded before. Each of these barcodes are pre-mixed, making that process simpler. They come as a kit, so you have all of the reagents you would need for barcoding, and we have a very straightforward user guide. A couple of considerations to note here. The palladium isotopes will be incompatible with other antibody isotopes in this range. For instance, if you're using 106 cadmium and 110 cadmium, that would not be compatible with the palladium kit. So you could either consider reducing the number of palladium barcodes you use, adjusting your panel design, or you could consider one of the alternative options we'll talk about in just a moment. Palladium covalently binds with proteins, so it labels all cells, meaning it is agnostic or universal. With palladium barcoding, the cells must be fixed and permed prior to barcoding. Thus, if you have fixed sensitive epitopes, you'll want to stain for these before the fixed and barcoding steps. I'll briefly review each of our application notes for these strategies. Here we stained PBMC samples from 4 healthy donors with 5 replicates from each for 3 different barcoding conditions. One condition included no barcoding and only surface staining with a 29 surface marker panel. The second condition was the pre-stain barcoding condition. This means that we barcoded samples, pulled them together, and then stained them with that same surface marker panel. In the third condition, we performed a post-staining barcode method. This means that we first stained with that same surface panel and then barcoded the samples and pulled them together. We did this to compare multiple aspects by these methods. So we compared the variability of population frequencies and intensities, the sample quality, effects on fixed sensitive epitopes, and the workflow efficiency. If we revisit our workflow slide, this experiment is using workflows 1 and 3, where we first barcode the samples and then surface stain, or where we surface stain and then barcode the samples. Barcoding reduced the population frequency and marker intensity variability by both barcoding methods. So this is demonstrated by significantly lower and tighter CVs in both of the barcoding conditions. The pre-stain barcoding strategy performed the best with the lowest CVs below 3.62%. And this is likely because these samples are stained and acquired together. The post-stain barcoding strategy was not quite as effective as the pre-stain strategy, but this still outperformed the no barcoding strategy. Now I'll move into talking about our CD45 barcoding option. So this barcoding strategy is based on CD45 antibodies. With this strategy, that means you're only barcoding immune cells. These CD45 antibodies are primarily conjugated to cadmium and platinum, but we do offer a few more options. The strategy is meant to be an alternative to the fixed cell palladium barcoding strategy. And essentially, we're just using antibodies here. So this is really a surface staining step, meaning it can be performed in live cells, hence the terminology live cell barcoding. This typically uses 7 isotopes, where you would choose 3 per sample and yield 35 barcoded samples. However, if you use all of the CD45 options we offer, you can achieve even more than 35 samples. Unlike the palladium kit, these antibodies come as individual antibodies, so you must mix the barcode on the day of staining. In the CD45 barcoding app note, we used the CD45 antibodies with a 7 isotope choose 3 approach. Here we used mixed metals, including cadmium, CD45 antibodies conjugated to cadmium, platinum, and yttrium isotopes. This was really a proof of concept study to show that CD45 live cell barcoding can be successfully performed and debarcoded. Here we used 1 large PBMC sample where this was split into half. So half of these PBMC cells received CD47 surface staining. And we did this as an intercontamination control, or an internal control, to assess intercontamination once we pull the samples together. So half of that PBMC sample received CD47 staining, while the other half did not receive CD47 staining. These were each aliquoted out into 35 individual samples, where the CD47 stained samples were the odd numbered samples. We barcoded each of these using the CD45 scheme and pooled those samples together for surface staining. So in this app note, we are using primarily workflow number 1, where we barcode samples and then perform the surface stain. To visualize all of the samples on a plot, we used Visney dimensionality reduction analysis. We did this with all 7 barcode channels, plus that CD47 antibody. And the Visney algorithm successfully identified 35 unique clusters. Each of these barcoded clusters correlated with the key as well as the isotopes used for barcoding. So we can see this here. For instance, if we take a look at the barcoded group 15, we can see that according to our key, its expression is correct. So we have labeled it with 89Y, 106 cadmium, and 198 platinum, while it's negative for the other isotopes that were not used for barcoding that sample. Additionally, this is an odd numbered sample, so we would expect it to have CD47 staining, which it successfully shows. So CD45 barcoding offers a great strategy to use for many applications, including live cell barcoding. Finally, we have the tellurium option, known as TMOL. With TMOL, the user must mix the barcodes and titrate. It does require some optimization. We're currently evaluating a more user-friendly, ready-to-go option. So if you are interested in TMOL, please do reach out. TMOL labels proteins, so this would be a cell agnostic or universal strategy. This can be a 7 isotopes, choose 3 to yield 35 barcoded samples. But if you combine it with other strategies, you can achieve much higher samples. For instance, adding just 2 platinum isotopes with a 9 choose 4 format yields 126 barcoded samples. The tellurium isotopes are in the range from 122 to 130 TE. These ranges do not overlap with antibody targets. So this means that this is not limiting your panel. You can still have a full 50+ antibody panel and include these 7 isotopes for barcoding. TMOL can be used on live or fixed cells. After you've acquired your samples by any of the barcoding strategies we've discussed, whether that is palladium, CD45, or TMOL barcoding, next you would debarcode your samples for analysis. The CyTOF software can be used to debarcode your samples. In our most recent software version and up, you can debarcode 100+ samples with any metal strategy. There are alternatives to the debarcoding with the CyTOF software that you may have come across, such as Premessa or ZunderLab, but with the newest release of this software, this negates the need to use alternative software strategies. Finally, you can always manually debarcode, but keep in mind this will likely be a lengthier process and can be subjective. So to summarize all that we have learned, CyTOF uniquely enables sample multiplexing. This significantly reduces your variability and increases workflow efficiency. These workflows and approaches are flexible. You can choose from multiple barcoding strategies that fit your experimental needs.